I'm Camryn Beaufort.
We worked with Dr. Schwartz on determining lead concentrations in the soil around Spartanburg as part of a larger project. So just to introduce the topic of what is lead, I'm sure everybody has heard of lead. It is a soft and malleable heavy metal with a relatively low melting point. This made it extremely favorable to work with in history. We could add it to lead pipes, you want to be able to transfer water, so you have something that you can easily manipulate and then you can move water around. So we also used it in gasoline as an additive and house paint as another additive, and it was just present in mining, metal production, and other things. But so now lead is pretty common, and it's because of human development. Lead isn't just around everywhere naturally, we put it around ourselves. We did the same thing with other heavy metals like cadmium and zinc. But starting in the 1970s, they started to ban lead from certain products like gasoline and house paint, but that was too late of an effort because we had been doing it for decades of using lead. And so lead became present in the soil because of deposition, and so now we can move this lead around when we move soil around, because it just stays in the soil, and when you move the dirt around through construction or just by general walking through it, it can end up in your house, it can end up pretty much anywhere. So lead does have a couple of risk factors, which is why it started to be banned in the 70s. Children are most at risk. There are many health impacts. It can decrease cognitive function, it can lower the IQ of children, it can increase ADHD presence in children, and there's some impacts on hearing, as well as many other health effects. There's been a lot of causal relations between lead concentration and just health problems like cardiovascular things and then just cognitive function in adults as well as children, but children are the most at risk. So the levels deemed safe by the EPA for children's play areas is less than 400 parts per million. That doesn't really mean anything, it's 400 milligrams per liter, but so they just have a scale, and so that would be kind of important. They also have similar guidelines for gardening, so around 400 ppm is also going to be conscientious of what you're gardening in that soil. So it's dangerous for children, it's dangerous for plants and animals, as well as adults, but you really want to be thinking about children because it will impact their whole life. So, and then there's this image. So our study is based on a project done by Dr. BR from the Spanish department, if you know her, and Dr. Brada, who works in environmental. So they were looking through parks in the Spartanburg area, all the way to Spartanburg County, and looking to see if there was a correlation between how good a park was and like what demographic of people lived in that area. So they analyzed 76 green spaces, and certain census tracts were found to be park deserts, and with certain minority populations, the parks in that area were just not as good of quality, the amenities were just not as good, and so now we were interested in finding out if there was a correlation between these minority park areas and lead exposure, because a lot of parks in this area, a lot of parks are built on reclaimed industrial land, so there is a concern about lead deposition in the soil and children being in those areas. So this is just kind of a repeat, like our question was, do these parks also have disparities in lead exposure as well as being with minorities versus like concentrated white areas? So we were hoping this semester to develop a method to measure lead in soil and determine whether or not there was a risk of high lead exposure in these areas.
So we measured the concentration of lead in soils here on Wofford's campus. We used Old Main, Green Hall, and Liberty Trail for our locations. We chose Old Main and Green Hall because we suspected that at some point they would have had some sort of lead paint, and then the Liberty Trail acted as our control. And so we followed the EPA Dustin soil sampling protocol, and then within this protocol were two sampling methods. One was the scoop method for softer soils, and then the other was the core method. And so we used the core method, and then in that method was just using an auger to penetrate harder soils, deeper soils, and then we went about a ½ inch deep and obtained each of our samples. They were each combined, taken 3 feet apart from one another, homogenized, and then dried in the oven at 40 degrees Celsius. Afterwards, we used a mortar and pestle to grind each of our soil samples to the consistency of sand, just to make sure they were all uniformed, and then afterwards, they were sieved to separate any rocks or larger sediments. And so for each location, we had 3 replicates, including the blank, and then 2 SRMs, standard reference materials, which was just what we used to compare our treatment groups to after our analysis. And so in this digestion method, we added nitric acid along with hydrogen peroxide and water to separate the lead from the soil. If we didn't use acid, we would not have been effective in analyzing the concentration of lead because the lead would not have separated completely from the soil samples. And that's the hot rock we use to heat our samples after the acid. This is the flame atomic absorption spectrometer that we use for our experiment to analyze each sample. Here we would place a capillary tube inside the sample and it would reach the chamber where it would be aerosolized, which just means that it would be converted into particles that would be able to be carried in air. Afterwards, it would reach the burner, which would atomize the sample, and then here would be our lead lamp, which was a lamp specific to the wavelength of lead, and that would pass through the atoms in the flame and the monochromator to separate the radiation of lead from any other radiation emitted. And so the amount of light absorbed by the atom is then detected by the detector, and then on the computer over there, it would read an absorbance, and then that's what we would use to calculate the concentration of lead in ppm. And then this is just the flame A on Wofford's campus. There is the capillary tube, the burner, and then the lamp is placed inside here and shoots the light across. And so here are our results. We predicted Maine would be the highest since it was the oldest, followed by Green and then Liberty. We can conclude that these results are not alarming because if you remember from earlier, the point of concern is closer to 400 ppm and none of our results exceed 55 ppm. And so when looking at our SRMs, the lead recovered, the expected lead recovered was close to or at 100%, which is what we expected, and so this tells us that our procedure is an appropriate one in determining the concentration of lead in soils.
So based on these results, we are going to continue with this research over the summer, and not us specifically, but over the summer this research will be continued in specifically using this method to study the park soil as we had hoped. So it will just begin with EPA digestion as we used before, and we are going to begin with soil taken from Dr. Bradman's house, actually, because she lives in a historic house in Spartanburg, and it is known that her house was painted with lead paint, and so we expect to find lead in there. We're going to analyze it against an SRM, again, just to make sure that our method is working, and based on those results, we will be able to just make sure that we can see actual high concentrations of lead in soil from Spartanburg, and then they will pick certain parks in the area, probably some from those higher white areas versus those higher minority areas, and then just see if there's a correlation between lead concentration and those disparities. These are just a couple acknowledgements for people who have helped us with our research. Maddie Glidden was our research partner in the beginning of the semester, but unfortunately she had to stop, but she will be continuing more in the summer. And then, of course, Dr. Bradham and Carson, our environmental partners, have helped us a lot along the way. And then, of course, Dr. VR for her work. And these last two are people that we met with from people who are working in Spartanburg to ensure that children in households that have lead are protected so they can fix the problem where it starts.
And also our mentor, Dr. Schwartz.
Of course. These are just some of the sources we used. We are open to any questions now. Thank you.
So if you do find higher lead levels in the soil in some of those parks, what could be done?
That's a great question. So a lot of the time it's just very difficult to remove lead from the soil, and unfortunately one of the solutions is just to cover it up. So in parks in other parts of the country that have been determined to be sort of dangerous for children, they have often covered it with another soil that is known to be pure, and they would cover that soil with mulch for children's protection. So as long as the lead is not at the top layer of the soil, children aren't at risk.
Another solution would also be to just cover it with concrete.
Are there other methods for doing this that don't require really nice equipment? Like are there home methods? Are there other, for folks who may be concerned about this if they live in historic areas? Is there anything they can do on their own?
Right, so that would be when you would contact people, Spartanburg Lead Safe. If this is a concern, you can call them. They mostly tend to work with people who have young children in the household just because they are the most at risk for lead poisoning. But that would just, you can call them and they will come and do an assessment of your house. They can do interior and exterior. We didn't exactly mention this, but for the EPA guidelines, one area that is very heavily analyzed is called the drip line, which is I think it's within a meter of your house, like along the wall. And so they check that soil, and because that's where people are most frequently walking. And so they will check that soil and see if there's a risk for lead.
Is there like any difference in results, I guess, between the different soil sampling methods? For sourcing?
So the scoop method was just for like softer soils. We suspected that since we're kind of mountainous, not really in the low country, we would use the core sampling method. So, yeah.
Just to kind of continue that, with the core sampling method as well as the spoon sampling method, you kind of take off the very top of it just because it moves around so much. But with the spoon sampling, it has been said that because that soil is so soft, it can be recycled very quickly, so that might lead to a difference in concentrations that you find.
I like that your research mainly focused on children, especially since, like you said, they have their whole lives in front of them, so their effects are much longer reaching. But I'm also curious how the effects of lead in the soil might affect the elderly, who also might be digging around in their backyard where they shouldn't be. How would that? What if
I think that's a very fair question. So I can't specifically say for the elderly, but with adults, I'm not sure what the age range on it is. Because the bones are more solid, that's where lead is stored, is within your bones. And so because children's bones are moving so much and deteriorating like at a different rate and like rebuilding themselves all the time, like they're more at risk because it moves around so much. With the elderly, it may be similar, but with adults, it has been found to have effects on like depression and just like, a lot of mental health qualities that you wouldn't really suspect there to be a correlation between lead and the way you're feeling.
All right, thank you.